Okay. The last lecture we did was on glycolysis. It's a very complicated process. Y'all tend to take it pretty well. Because glycolysis is followed by respiration or followed by fermentation. This is not quite as complex because there's no, if you don't have this, you do this. This is straightforward. All plants do this. Some of you single-cell animals, Volvox does it, amoeba does not. Botticelli does. If it's green, probably does. But that's chlorophyll. And what I have here is just simply a start off saying the sun's energy drives the process. Carbon dioxide enters the process. Chlorophyll makes it happen. And in carrying out this, oxygen is given off and glucose is produced. Now, that's a very simple way of saying it, because you also get produced such things as sugar and sugar cane, and you get the starches like in a potato and a carrot and things like that. So let's just go through it. I basically just said that. I mean, plants do it, some bacteria, some protists. They use the energy of sunlight to make it happen. And that is used to produce, I didn't say glucose, did I? Sugar. Because a lot of things produce, not just glucose, but they're all carbs. The only carb we can use is glucose. So when you eat that potato, you change that starch to glucose, then you're going to use the glucose. So you haven't got to eat glucose right off the bat. You can eat something akin to it and then change it to glucose. This is what you do, basically. And sometimes they're fattening because there are a lot of starches, there are a lot of glucoses in a starch. So when you eat, a lot of mashed potatoes, that's a lot of starches. That's more glucose than you probably need to stay alive. And the body will save the rest of it as fat reserves, and you'll get fatter. That's why eating too much does to you. So leaf pigments now, there's xanthophyll from the last class. There's xanthophyll, there's all kinds of, there's keratin, there's all kinds of pigments. All of them use sunlight to make this happen. But the one that's the most often user is chlorophyll. There's an A and a B. And A uses one wavelength of light, and B uses another wavelength of light. That way everything's covered. It's pretty efficient, everything's covered. If you had just A with this wavelength of light, then the other wavelength might not be used, and that's wasteful. So plants cover it. So it's the energy that's captured. And what's keeping you alive right now, your energy, it came from the sun. That's all there is to it. Everything you consume comes from the sun, whether it's the cow you ate that ate the hay, or whether it's the black eyed peas you ate that directly came from. So you can't get away from sunlight. It uses water. You got to put in water, and you got to put in carbon dioxide, and you got to put in sunlight and make it happen. And when you get done, it's going to release oxygen. That's trash to a plant. Ain't trash to me though, buddy. I need that oxygen. The carbon dioxide the plant needs is my trash that I get rid of, and the plant it needs that carbon dioxide. The food you consume, whether it's peas or snap beans or corn or potatoes or sweet potatoes or squash or okra, anything you want to think of, that's our food. Those are producers. Producers mean they produce carbohydrates that animals live off of. The overall reaction simply is the things you can see. I need 6 of these waters. I need 6 of these carbon dioxides to make one of these. You see that 6 right there? I need 6 to get my 6 C's. 5 won't be enough. It ain't C5s. And if you add these numbers up, I got 12 H's on this side and I got 12 over here. They're just reshuffled and rearranged. And if you look at the O's, what's 6 times 1? 6. What's 6 times 2? 6 times 2, 12. But what is 6 plus 12? Oh, the 18 over here? 6, what is this? 6 times 2 is, and that 6 is, they're all accounted for. They went in like this, and came out like that. That's what we have, that's what we live on right there. That's our gasoline right here. We need this too. We need this so that we can take this substance and destroy it and get energy. Then we give this back out. We breathe out what the plant took in. It's the exact opposite. When I showed you respiration, I showed you the sugar plus the 6 oxygens, giving off the 6 waters, and 6. You're just simply undoing what the plant did. It never ends. It's a cycle. Of course, now if you lose your plants, you break the cycle and we die. Do plants need us?
Yes.
Yeah, for the oxygen. They need us too. So we're in this world together. Leaves and their structure. A leaf really is a solar panel. It's a solar collector. All it's for is designed to. You can take a leaf and put a plant right there and leave it alone and come back. All those leaves will turn to face the sun. They'll grow that way. And you can spin that sucker around and have them facing in here and then forget about it. And next thing you look, not today of course, but they'll all be turned back around. Now there's no nerves in a plant. The plant can't see that sunlight. The question is, how does he know which way the sun's coming in if he can't see? That is called phototropism. It's the actual sunlight itself. If one side of a stem grows faster than the other side, it's going to make the stem spin around on it. If I got my 2 fingers, if this finger grows faster than that does, it's going to curve this way. Phototropism just determines how fast the side grows, and that will actually make that plant leaf turn and again face the sun. If you have real plants in your home, you got to spin them. But they'll become one-sided so bad it ain't funny. A tree outside in nature, the sun goes across the top, and it got to be spinning. The sun faces in all directions. But in your home, or in that window there, those leaves are going to point themselves toward that window. And it's going to grow that direction, and after about a year, that sucker is so lopsided you can't do nothing with it. So keep spinning, keep spinning. All right, so the leaf is a photocollector, solar collector, full of those photosynthetic cells that we call mesophyll. That picture you drew, all the mesophyll. You got the palisade mesophyll. What's the other kind? You remember that kind? It's the S word. It's a common word. Like in your kitchen, you have one there. And you wash with it. Spongy mesophyll. There's palisade mesophyll, which is like books lined up in a shelf. That's on the top of the leaf. And you got the other kind kind of stuck in there in all directions like a sponge. That's your spongy mesophyll. Well, the mesophyll, that's the photosynthetic cells. That's green. Now, the cells on the surface, they're basically nothing inside those because light shines right through them to hit the cells under them. The raw materials means, what do I need to make something? What are the raw materials of a ham sandwich? I need the bread, I need the ham, I need the mayonnaise, my lettuce. So raw materials are what you got to have to make something happen. The product, the sandwich, would be the sugar, which is usually, I can't even say that. It's just sugar. We'll change it to glucose when we eat it, because they're all the same chemical. They're all the same atoms. We just rearrange it. And oxygen is given off, and like I said, animals, they've got to have oxygen. I told you earlier, there's some things that would die if you breathe on them. Not call you breath either, because they can't stand the oxygen. They're the obligate anaerobes. Yeast is one of them. They will not live with oxygen, but that's why they do their fermentation routine. Now, here's a picture If you got the PowerPoint, you got the picture already. And I talked about the mesophyll, and they're lined up like books on a shelf. This is the top. This is the upper portion of the leaf. As you see here, the epidermis, there's nothing really inside there. There's no green little dots. It's just a covering. And of course, the cuticle covers this. You ever felt a magnolia leaf, how thick it feels? How firm it feels? The cuticle is waxy. And a plant that's supposed to grow in direct sunlight has a nice thick cuticle to keep the water inside the plant. Plants that grow in the shade are real delicate, they're real soft. They don't need that big cuticle because they don't get the sun directly on them. When you buy a plant and you're going to plant your house, you need to read the directions, say, well, you want full sunlight, filtered sunlight, or whatever. You plant in the wrong place, it ain't going to survive. And if you take a plant that needs full sunlight and you put it like up in the woods behind your house, in the shade, you might find that you have to buy a new plant for them. So be sure to read where to put it at. These in here, just slapped all around, those are mesophyll, but that's the spongy. And there's air spaces in here. This is air. They're not crammed packed, there's air. This is a stoma. This is a stoma. And these two guard cells, when they swell bigger, they will shut the stoma. Or they shrink to open the stoma. And that's how the air, the carbon dioxide goes in, and how the oxygen goes out. And some leaves, not many, some leaves have stomata, that means 1,000 on top. But usually, if I'm going to look for a stoma, I'm going to look for one to be on the bottom. Because these gases, first off, the gases can't get through here to get out anyway. These like books, they're crammed together. So the way the gases get out is through the air spaces, and that's why the stomata are on the bottom. One's a stoma, two would be called stomata. Now don't get stoma confused with stroma. You put an R in there, you got a whole new beast. Totally different. A lot of folks got one R stuck in there will make the answers wrong. So you gotta watch out. Now, so these are the palisade. And I think of a palace where the arches go up and down and like a palace. That's what they're called, palisade mesophyll. This is the leaf bundle. This is where you find the water flowing. Xylem carries water. Phloem, and I think at the F sound, carries food. If I remember what phloem does, food, then by default, I know that he must be doing water. He's always on the surface. You're never going to find him reversed. Nature just decides to put xylem always on top, because and when you cut a leaf off, it's going to bleed. It's going to get liquid. Some of your, I've seen a lot of folks chew on something. The lower epidermis, as you can see, has nothing inside it to help do photosynthesis. There's a cuticle on it also. Whether thick or thin depends on where it lives. And whether you have, now I don't care what kind of leaf you have, whether it's a monocot or a dicot, but have some degree looking like this. This leaf right here, where the veins go in different directions, had to be a dicot. Why did I say that for? How do veins run in a monocot? They run up and down. Think of a blade of grass. Think of a piece of corn. Corn doesn't do that number there like oak tree does. So if you see a picture of a leaf, and there's a vein, 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 they're running up and down, aren't they? So what leaf would that be? Monocot or dicot?
Monocot.
That's the monocot. And if you see one and the veins are kind of oblong, they're not round. If you take this, if you cut a pencil perfectly, it's going to be round, right? If you cut at an angle, it's going to be oblong, right? So if these veins run right at an angle through that leaf, and when I cut it, they're going to be oblong shaped. If they run right down that leaf, perfectly parallel to each other, they're going to be round in shape. So I can look at the shape of the vein and figure out, this is a monocot. just by, is it round? Now, if I see round, 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 I know they're going up and down that leaf. If I see oblong and oblong, and they were cut going this way, like the palm, like your fingers, and now I know that would be dicot. Now, I tell you what, though, and I'm telling you this, that's high level biology. A person knows that has been using a scope for years like I've been using one. There's some things I see, and I just know this is, I know what this is because I know the way things are cut. So, this is the vein. This is the vein. Some folks will call this the vascular tissue, because it is vascular tissue. Those right there, that hold things together, that's called the vascular sheath. Now, it's not named, but it's like a knife sheath. You put your knife in a sheath. Some folks will call this the vascular bundle. Depends on what book you read. And that problem is there are so many names for the same thing. And when you get educated, you start to realize that there are a lot more names of this thing than just vein. Remember, vein probably is a kindergarten word. I'd rather say vascular bundle than vein. Because I got veins. But then again, my veins are called vascular tissue too. Then you got the cardiovascular system in your body. So any vein, any vessel is something called vascular. So anyway, I featured this one other stoma with its two guard cells that open and close it. I featured the mesophyll cells, both kinds of them. Actually, they're the same color. They're colored here just for a teaching device. They wouldn't be different colors. When you did the lab, when you crushed the leaves and let things come that paper, you were letting these things out. Those right there, you were crushing those and letting out the chlorophyll. But you also let some yellow stain. You saw yellow creep up too. So there's a lot more in there. Okay. This is basically, I think most kids know this. Water enters the root and it travels up to the leaves where it's going to be used to carry out photosynthesis by those things called xylem. The carbon dioxide and the oxygen, they cannot go through that cuticle. They must go through the opening that we call the stoma. One's a stoma. 5 openings you would say are not stomas, you would call them stomata. Okay? So stomata would be a lot of one stoma. It means hole in Greek, which makes, that's good, it's a whole leaf. Most of these terms, though they might seem weird, they have a common name, and when you hear it, that makes perfect sense. Hole, that's what it is. It's a hole in the bottom of the leaf. There are many holes, though. 2 guard cells that flank it on both sides, they open. When they get fat, they close. When they lose water, they shrink. And that's what opens and closes the opening to let oxygen go in and go out, and carbon dioxide go in and go out. These are some pictures. Now, this is a person's drawing. This is for real, and that is for real. This is someone's sketch of what they thought it looked like. These are 3 views of stomata. And every one of them, the guard cell is always longitudinal. It's never like this. This is the guard cell. That's the guard cell. Matter of fact, that's the guard cell right there. That's the guard cell. And that's the guard cell. That's the guard cell. If you look right here, this is the guard cell. And that's the guard cell. And this is the guard cell. That's the guard cell. And that is the opening that's called the stoma. If he gets fat, then the opening closes. If he gets skinny, the opening opens. And that's what lets air pass. Now, before I tell you, are you looking at the upper surface or the bottom surface of the leaf?
Bottom.
Huh?
Bottom.
Bottom. I mean, there's 2 answers, but the best one would be bottom. They might be on the top, but I know they're on the bottom, so I would answer bottom. And you see this many, if this were a top view, you might see 1 and no more. But this many stomata, this many stomata, that one right there, you probably couldn't guess if I'm looking top or bottom. That's only 1. But when you see this number of them, you go, well, that's going to be a bottom there. That's going to be the lower surface. Okay? Now, It's the light now. Does a plant need sunlight or just light? Huh? Just light. Just light. I mean, the supply rooms have no windows. If I left the lights on, though, I can go plant in there. They just need light. Not just, sun's good. They just need light. Just need light. And of course, you know whether you see the red light or the blue light or the green light, and based on what frequency is being reflected. What is absorbed, you never see because it never comes back to your eye. So if you see on the screen red, anybody got a laser? Take it out, shine the screen. I want you to shine your laser at the white part of the screen.
Maybe not.
At the red part and at the black part. You don't have it?
Not on me.
Well, wait, you just have one here? Some of them have lasers. Or I don't use it enough to find a laser on here. I don't see a hole. But what I'm telling you is that if you took a laser and shined on here, it'd be real bright right here. You know about we be real bright for? What's white do? Reflect or absorb light?
Reflect.
It reflects light. If you let your laser beam hit that black, it's going to get real dim. Why real dim? Because the light's been absorbed. If you shine the laser on the red, what's going to happen then, you reckon? It's going to be bright also. You know why, don't you? If you see the red, it's been reflected. If you shine that laser on anything black, you can find that your laser beam is not quite as bright because the black object is absorbing it. And I learned that from another lab. I was pointing down, and every time I pointed something black, my laser would turn off. It wasn't turned off. It was being absorbed by the black word. So now I know that to shine under the black and not actually on the black. Okay. Some simple physics here because we need to understand these terms. This distance is the same as that distance. I can measure from top to top or bottom to bottom, and this wavelength would be, if that's 5, I'll guarantee you that's 5. How fast it goes is something else entirely. So peak to peak or trough to trough is the wavelength. Now, if this were further apart, if the wavelength, say it's 5, say it was 10, let's say I stretched that spring out. If that light had a higher wavelength, would it have more energy potential or less? Big screen. If you had a longer wavelength, would it have more energy in the light or less energy in the light? It'd be less. Because, see, it's inversely proportional. As a wavelength increases, the potential energy decreases. If it's conversely proportional, what would that mean? As one increased, the other would too. But to be inverse, you got to think opposite. As one increases, the other one will decrease. And this is inversely. So if I make those closer together, would the energy be more or less then? If they're closer, the energy would do what? It would increase. If I decrease wavelength, that's going to increase the energy. Red light versus purple light. Red light has real long wavelengths. Purple light has short ones. There is more energy stored in purple light with those short wavelengths than in red light with the long ones. That's why in a dark room they'll use a red light. In a submarine, a red light. Because those long wavelengths don't interfere with your night vision. They don't use purple lights in those places. They don't use anything but red, and red's there for a reason. I understand now that the lasers, All my weapons at the house have red lasers on them. They're now making a green laser beam that would not be soaked up like this, but they cost a lot of money. Maybe in years to come they'll be down to Bobby Tep could afford. But at night it's nice if I hear a noise and I got my pistol. I can't see you. I just shine that laser. That thing hits that assailant, he's gonna run probably because what's he know? There's a gun pointing at him. So most times the laser itself often allows you not to have to pull that trigger because you got. And now my brother told me this flat out. If you're in his house, he's going to kill you. No questions asked. I ain't doing that. I've been in Vietnam. I've killed folks. I don't want to do that ever again. So if you're in my house and you're not threatening me or my family, if you got my brand new TV in your hand, you might be in trouble. I'll say, put it down carefully. If you drop it, now, and I'll say, stand there. And I'll call 911. Now, if he were to try to walk out my door, I'm sorry, man. I don't think I'd shoot the man. I sure ain't gonna wound him, because then you're gonna sue you back, right? So, I've never been in a situation where I've had to decide on being just a mean guy. I don't think I would shoot somebody unless he threatened me or my wife or somebody who was with me. I don't think I'd do that. Now, if you put my new TV set, I don't know, I might not do it then. Because would you get life? Would you get life for stealing a TV? Would you get life? Would you get death for stealing a TV?
No.
So if I shot you, I better be scared for my life. But down in Florida, there's a new law now. It's called Stand Your Ground law. It used to be where they'd approach you, you had to back off. You had to run for your life. Now they say, if he approaches you and gets in your space and you think you're threatened, shoot that rascal. There's now a case pending right now in Florida where a man did that, and right now he's probably going to be let free.
Is that the one that had the laser?
He'd already attacked that 91-year-old woman, and he'd attacked her and knocked her down. And the guy pulled a gun out and said, stop. Well, the guy he's a drunk, began to approach him. Instead of him running, he shot the guy dead. And I can't say I blame him. He didn't prove he'd do body harm to you.
Well, it was another issue where a guy came back from the war. Somebody broke in his house and he had a weapon and it had a laser. And the person started to run and he shot him. And I don't think they charged him for that.
Someone told me one time, a police officer who I went to school with says, if you ever shoot somebody, drag him back into your house. That he said, he's got to be in your castle before you can even make a defense. If he's outside your front yard, he's running from you, he's no longer a threat, you better not shoot him. I have a carry license in my wallet because I'm one-legged. And when I go hunting my four-wheeler, you could tell me I'll take my four-wheeler. I always have my 45 on my side or my 38 when I'm out in the woods somewhere because I might see a wild dog. And I got a license to carry, but I don't want to shoot anybody. But if I get stopped by the law, they say in my car is my home.
Right.
We can lay in there also. I don't want my gun here and the bullets in his trunk either.
Right.
What good is that?
Yeah.
And so I have the permit, but the permit I can't carry here at college, that kind of stuff. But when I go on the road, if I go to visit somebody, now I know that Alabama, you go across the southeast, Alabama will recognize Georgia's permit. When you get to Texas, past Texas, they don't recognize permits anymore. So if you plan a motorcycle trip across country, and you got your gun with you, sometimes you can put on your side, sometimes you can't. I said when you get stopped by the law, I'll tell the guy if I got stopped, I said, I'm armed. He says, just keep it out of sight. And then I always give him three things. Well, I give him two. I give him a license and my carry permit. And I don't say anything more. And then he says, where's it at? I says, under my seat. He says, keep it there. That's all he said to me. And I got a warning for almost speeding, but because I was respectful to him, he didn't come back. And he could have given me a ticket. But I found that you catch more flies with sugar than vinegar. So I was nice to him, man. He was nice to me. He was late. All right. Shorter wavelengths, like I said, the purples and violets, they they have a nice energy content. If you get longer than red, you're called infrared. And when you get higher than violet, you're called ultra, and you can't see either one of them. Well, the black light, you can see ultraviolet for us. But with your eyes, you can't. I hear deer can see higher, though. The orange vest we wear, that to me stands out like a beacon. Deer doesn't register because his spectrum of light is not the same as ours. I mean, he could run to the woods dead of night, full blast, and not hit a tree. And when he sees something really good, I wouldn't even try that. Now this is the spectrum here. And I got it ranged from short wavelengths to long wavelengths. You get longer, you're infrared. You get even shorter, you're ultraviolet. Here's how it works. If this is the spectrum, from here to here, X-rays that we take through your body, because you can't see them. Ultraviolet light from the sun is right here, but you cannot see, but you sure feel that sucker after a while. Above this side are your infrareds. You know your radio signals when you listen to radio? It's also on this side. Walkie-talkies, where if you're going to start your radio station, they give you a frequency to be in. And that frequency is yours. Now, that might be nothing like in California, like your frequency, but you won't interfere, you're so far apart. But if you have frequency 54 here in Columbus, I want to start one in City 54, we got a problem. You can hear both stations come to your radio, so that's licensed by the government, of course. These are nanometers, and if I took a meter stick about that long, I divide it into 1 billion sections, one of those is a nanometer. If I did 1,000 sections, one is a millimeter. 100 sections, which would be about that long, that would be a...
Centimeter.
Oh no, that'd be a decameter. Centimeter about that long. But you're right though. I said long. Alright. Remember the word Roy G. Biv? Roy G. Biv. It goes red, orange, yellow, green, blue, indigo, and violet. The name Roy G. Biv is the spectrum backwards. It's backwards. So when I write down Roy G. Biv, I got to say blue, I can't, violet, indigo, blue, green, Roy, yellow, orange, red. I'm reading backwards. I'm being dyslexic. But I've heard Roy G. Biv, I guess, as long as I can remember. It ain't mine. Somebody ahead of me thought that up. But right here, there's your greens. Now here's my question to you. Is a plant using green, or is he not using green to make food?
Not.
Why you say not? You're right. Why?
Because it reflects
It's being
the light.
reflected. It's being reflected. What's being soaked up is what he is using. The blue is being used. The violet's being used. Now, and right now, when the kids, we collect those leaves, right? We have the yellow leaves, has the red leaves when spring came, and there's still greens out there. As those leaves change colors, say there's more pigment than just chlorophyll in there. Some pigment is going to be using green. When the color changes and that thing turns orange, is he using orange or is he not using orange? That particular pigment. He's not. Is he using green or not? Yes, he is. So all the colors get to be used because they have all the different pigments. But because it has so much chlorophyll, the leaf looks to be what color? And he's not using green. But now the question might be, is green light ever used to make photosynthesis? Absolutely. The xanthophyll is yellow. Yellow's being reflected, so guess what's being used? The green. The keratin is yellow, is orange. That's being reflected, so what's being used? All the other colors. Now, I ask this question to you. Of all of Roy G. Biv, which one do you think is used the least by photosynthesis? And you would say what?
Violet.
Used the least for photosynthesis.
Green.
Because it's the one being reflected. If you get reflected, you can't go in, can you? So of all the colors you see right here, the one that is used the least is the one that you can see, the green leaf. Now in autumn, when that green goes away and the red that's been there all the time shows itself, but then you're not using red, then you are using green. That proves that leaf does use green, but chlorophyll is not using green. Okay? Now, the photosynthesis uses wavelengths because those wavelengths can make electrons be generated. It's the electrons. One more time, it's the electrons that's going to make things happen. Remember in respiration how the electrons sent the hydrogen out and hydrogen came back, but the electrons drove them out so they could come back. Well, here electrons are going to drive this too. It's going to drive this too. Electrons are, we use them for light. Plants and everything does also. All right. Pigment absorbs light energy. So if the thing is green, he's not using green, is he? If it's red, he's not using red. He's absorbing what he's not using. And I have C on the next page. There are three things that are possible. One, the energy is going to be wasted. It's going to be lost as heat. It's going to be wasted, not used. Two, it's going to be changed to a longer wavelength. If it comes in as purple, which is short, if it changes to longer, maybe it become green or become yellow maybe. So it's going to go to a longer wavelength and the color is going to change. You heard of fluorescence, how your watch will glow in the night? I know the Army didn't use them because they'd be visible, but you can shine a light on and take it back off and you can see the thing. That's fluorescence. And what's happening, the light you shine on that chemical, it goes in there as white light, but it is moved to a longer wavelength, green, and it glows as green for a little while. Even the oceans do that. You ever seen the bioluminescent ocean? The red, how it glows at night, almost like the aurora borealis. There's some strange things happening just because the energy can be emitted as a longer wavelength because you've lost some power. If you go in there, short wavelengths, short wavelengths, there's your peaks, and you change a longer wavelength, then you lost energy, haven't you? And you've also changed your color too. That's your fluorescence. And the next thing is what plants do. You can use that to make a chemical reaction occur. That's what they do. They make a chemical reaction occur. In chlorophyll and the other pigments, that's what starts the reaction happening. Because the chlorophyll releases an electron, and the electron does the rest. So like start dominoes falling. I get one to fall, the rest is going to go. If I can make that chlorophyll release one electron, it's going to happen. The light makes it happen. Now, the light hits the chloroplast, but there are proteins in the chloroplast, and the proteins is what really makes it happen. They're the ones found in chlorophyll. They're in the cell membrane. They're in the membrane. Remember how the electron transport chain was in that membrane? These are in the membrane of the chloroplast. Now, prokaryotes, you have eukaryotes and you have prokaryotes. Do you remember the difference in the two of them? Which one has a nice, pretty nucleus, and which one has a nice, pretty vacuole, all-in membranes? The eukaryotes. Now, another one they have the chromosomes, but not in a little bag, because they're floating around inside there. That's the prokaryote. And the eukaryote has the chloroplast, because that's the membrane. A prokaryote has no chloroplast. Their chlorophyll just floats around. It's not contained in a little bag that we have named a chloroplast. They're there. And most times in a prokaryote, the chlorophyll is attached right under the cell membrane. You know why? Who gets more light, deep inside or the surface? Well, he wants to be near the surface, doesn't he? So the chlorophyll for a prokaryote is usually right under the cell membrane, close to the surface, so when light shines it, he gets it. If he's deep in the middle of that cell, he might find that he's in the dark. So, but what do you think is more efficient? The way a eukaryote does it or the way a prokaryote does it? Eukaryote. First off, they're allowed to have more of the chloroplast. They're not just centered. And remember that leaf I showed you earlier? They're all near the surface. And the sun shine on top of a leaf or bottom of a leaf? So where do you find those mesophyll cells trying to get to? That's why they're all packed together on top. And then the spongy ones, they're not locked together. They're on the bottom, but then again, the bottom of the cell doesn't get much. It gets ambient light. Because even under here, I can still see that, can I? It's not being shined on. That's ambient light. Because there's a leaf under here. It's going to hit it and bounce up, right? So I can use chlorophyll on the bottom, but the thing about it is, light shines on the leaf surface. And that's where you're going to find the palisade cells all trying to get there, and they're all packed together, because that's where the light is. Okay? And these two types, they're prokaryotes, and I'll get it later on with you. Both are kinds of bacteria, because bacteria are prokaryotes. Okay, chlorophyll and other pigments. Now, there are other things than chlorophyll. They're called accessory, because chlorophyll, he's the one carrying the load. He got helpers, but he's carrying the load. First off, any substance that absorbs light is a pigment. But the color of the pigment is what it did not absorb. So if you're green, you're a pigment because you absorb purple, you absorb violet, you absorb yellow, you absorb red, but you did not absorb green. You're a pigment. And like I said, all of your photosynthetic cells have chlorophyll. If they have anything at all, they got chlorophyll. Okay? And chlorophyll, like you know now, it can work with anything but green. It can use all the light colors but green, because green's the one that don't get in because it's reflected back. And if you see it, it didn't get used by the cell, because that's why it's green. Black pigments absorb everything. That's why they're black. White pigments reflect everything. The question is, if he's reflecting everything, why do you call him a pigment? By definition, what's a pigment? He absorbs something, right? Well, white paint. Why is that paint white? Because every light is being reflected. Well, why call it a pigment then? That's a misnomer. Because if you're white, then you're reflecting everything. And even here it says reflect all or most. And the problem is, if you reflect all, I'm not sure you're really a pigment. Because the definition says any substance that absorbs light in any way is a pigment. Well, if you reflect all the light hitting you, I ain't sure I can call you a pigment then. You follow me? But we do, because when we discuss pigments, we're discussing basic. Well, I say pigment, I'm thinking color. If I want to dye my shirt red, I want a red pigment, right? I want the one that's going to be reflected. If I want to dye it blue, I want the blue pigment. Why is he blue? Because that's when he's not absorbing. And why is he a pigment? Because he's soaking up the yellow, he's soaking up the green, he's soaking up everything but the color I wanted. So I still have a problem, and it's just me, Bobby Tess, with a white paint being called a pigment, only because there's no light being absorbed, it's all being reflected. But that's just probably just because I'm just pinching pennies. Not that it really matters. Pigments have their own absorption pattern, and you're going to see some colors in a minute because different pigments absorb different ways. Now, this is a picture you have on your that I gave you. 1, 2, 3, 4, 5 kinds of pigments. And now, if you're down here, this is the color I see. These colors I'm not seeing. You know why I'm not seeing these colors? They're being soaked up by the pigment. So let's talk about keratin. Now, what color do you know keratin to really be? Orange. Now, at this wavelength, keratin is being absorbed. Now, that's your max absorption, right? It drops off, comes back down again. Look, now he's visible. So in the 550 wavelength, this pigment can't do any good for you. And at 450, yeah, he's soaking things up. This color light here is green. So if you look where it crosses, he's soaking up green. This is blue. He is soaking up. Looks like he gets more blue than anything else, doesn't he? See how high it goes up there? That's on the same line blue is. So of all the light that hits keratin, which one is he soaking up the most? Look at the top, look at the peak. And what's that peak coincide with? Look down the bottom of it. Ain't that his high point right there for keratin? Now look down here at the bottom and say what color would that represent? Blue. Now, what if you look here and you look here? About equal, right? He soaks up at this wavelength as much violet as he does over here green. Is he soaking up green? Yeah, he's getting some of it. Now he's going to turn back and look yellow. Look orange. Why is he looking orange?
Abstract.
He's reflecting it. He's not soaking it up. And that's why carrots are orange. Now if you look at chlorophyll A, let me ask you a question. What two colors does it most absorb? Find the points. What two colors does it most absorb? Now there's chlorophyll A right there. Now read straight down. What color is this corresponding to? What would you call that?
Violet blue.
Bluish violet or violet blue. Now look over here though. He's also absorbing a lot of this color, which is reddish,
Green.
not red.
Green.
Now, which color does he not absorb? He's hitting zero, isn't he? But that's your green. That's why you see green. Now look at chlorophyll B. He's peeking out of what color? Not this one. He peeks out of what color? Plain old blue. Do these two chlorophylls get in each other's way? They're peeking out different colors, aren't they? Now, does he have another peak somewhere else on this line? Follow it. He's green here, right? Follow it back up. Oh, he's peeking right there. At what color? Which one would be the brightest red? Chlorophyll A or chlorophyll B? Now this one's A and this one's B. Now this one's almost toward yellow, isn't it? Which one would be the most vivid red? This one, which is A, or that one, which is B. Now look at my chart down here. I'm going this way, right? I'm going from yellow to red to darker red. So between those two, which one uses that nice, dark, heavy red light? Yeah, A does. And which one is using that kind of a lightish color red, kind of yellowish red? That's how you read that. Now look at this one called phycoerythrin. Phycoerythrin. Now, let me ask you a color on this one here. What color do you think that comes off as when you look at it? Red. There's phycoerythrin. Going across. Actually, it's kind of yellowish. And over here on this side, certain times it's going to look blue. It's always going to absorb light in the green-yellow spectrum. While this thing is kicking off, he's absorbing it. Look at phycocyanin. Phycocyanin, that's this one right here. If you buy cyanin ink for your printer, what color is that?
Blue.
That's blue. Now look what's happening. He is absorbing tons of what color? Tons of what color? Yellowish red. He is reflecting tons of bluish green. If you ever look at it, it is bluish green. It ain't pretty blue. It's a bluish green color called cyan. Erythrin is the Latin word for red. Phyco means plant. So this thing here is absorbing a lot of the light in the yellow-red spectrum. This is actually a misnomer. This is a good name because he's reflecting the blue light. Keratin's a good name because, as you know, carrots are orange, and he's reflecting orange light. When you get above this, they can't use it. It's not reflecting it, it's not being used. This is being reflected, reflected, reflected. That's just gone. That's just gone. So, if I talk about chlorophyll A, I got a question for you. Can he use any light above the 700 wavelength range? Chlorophyll A, that's the pretty blue one. Can he use light above 700? Probably not. Can he use any light above 650? Oh yeah. Now look at this one here. Where they cross. They're using what? 650 range is red. Are you seeing red? No. They're soaking red up. And the higher you go, the more they soak up. And that's why these, of all the pigments, which one has the potential to soak up the most light of any color? Of all the pigments, which one has the most potential to soak up the most light? Chlorophyll, which one?
B.
B. Who would be second, maybe tied with third? Nuh-uh. These two, they get more of their color. I think he's tad higher though. So it'd be first place, second place, third place. Now between the keratin and chlorophyll A, which one do you think is in the next place?
Keratin.
Keratin's right here, topping out. You didn't go higher than that. That's just top right there, buddy. But how many of them peak twice in their career? He'll peak twice. This one will peak once, twice. He'll peak once, twice, 3, 4 times. A plant uses all of these because he needs to absorb that light to make the process go. The light reflected, you can't use that light. And because there's so much chlorophyll in the plant, there's so much of it, that's why the leaflet's green. If they're like in squash, what color is squash most times?
Yellow.
Yellowish. A plum, how do you know a plum is ripe? It turns what color? Purplish red. What color is it when it's not ready yet? Green. Green plums are bitter. Pick blackberries. Which ones do you not pick? The green ones or the black ones? The green ones are still growing. They're going to turn black eventually, but the green, because they still got chlorophyll working, they're still making themselves. And when they get really ripe, they'll fall off the tree, hit the ground. They'll rot. New seed goes down, new blackberry bush comes up. Fruits actually are made to reproduce the plant. Of course, I like the cherry, I like the orange, I like the apple. But don't you dare eat apple seed. Apple seeds are poisonous. Apple seeds are poisonous. Did you know that?
Apple what?
Apple seeds. The seed in the apple is poisonous. Read about it. Read what's in that sucker. I just see chomp, 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 chomp.
Isn't it cyanide that's in it?
How many folks, they don't eat the seeds? And if you did, you're going to get sick from some mysterious reason and might blame it on the oyster you ate.
Or your wife?
Yeah. But you don't want to, don't look back there. You get them daggers? All right. There are a lot of things, and I was doing research on the apple just by chance. I read that every part of a rose is edible. Every part. I went, ooh, let me check on other things, and I found out for the apple, the core is good, the rind is good. A lot of folks peel it. But they said those seeds will mess you up. I think that's so that when the apple falls to the ground, animals will leave it alone and let that tree propagate itself. Anyway, that's just some food for thought. Here's another way of looking at it. And this is another source. And as you can see, they are very similar. Still, chlorophyll B beats everything. Keratin comes in. You compare these two, keratin beats them. Keratin beats them still. I don't like this one because I don't get to see my other colors. So if I took off this, I'm going to have a lot of area here not being used. And you will think in this picture there's nothing using them. But there is. There's those other two products. But if you look at this design, why do you reckon more light is used from this side than that side? Why do you reckon more light is used in the 400 to 500 range compared to the 600, 700 range? Who has more energy in it? The 400 light or the 700 light? Who's got more energy?
So,
The closer you are together, the more. So if you're 400, you're close. 700, you're almost twice as far. They're going to get the goodie out of good. This light has more energy in it. Therefore, these pigments are going to use all they can. This light has less energy. They'll get some of it. But if you look at this spectrum right here, there's more energy in this light and less in this light. And that makes sense to have the pigment more suited for the high energy light. If it were flipped backwards, that'd be pretty inefficient, would you think? I mean, if this was the high side, I'm going, man, they're not using all this potential over here. But the way it is, they do use that potential. But right in through here, where the light's kind of medium strong, oh, they got something getting it. They got something getting it. But, if I showed this picture only, you would not think that these even exist. And if you look carefully, a lot of light is being gotten. A lot of light is being gotten. All right? Now I want to talk about the chloroplast, the actual organ of photosynthesis. Inside the chloroplast, you don't find cristae. They're called thylakoids. Okay? Because if you think about it, you know that the mitochondrion is the organelle that does respiration. But really and truly, it's the cristae inside the mitochondrion that's doing it. So in this case, the chloroplast is the organ of photosynthesis. But inside that chloroplast, those membranes you're going to see in a minute are called thylakoids, and they're the ones which are doing the actual photosynthesis. Both of them have them. Both of these have them. They have the chemicals. But only eukaryotes have the membranes. The chloroplast would not be in him. They would just have the chemicals floating around outside the chloroplast, near the cell membrane. And I'll show you a picture. It's going to make more sense when you have the picture yourself. But they're stacked like a bunch of pancakes, like you go to IHOP and they stack them up on your plate, and that's how one doesn't really actually touch the other. They're just stacked up that way. Mitochondria have two membranes. The outer one, as you saw, and the one inside. They have the mitochondrial membrane and inner membrane is called the crystal. This bad boy's got three membranes. Three of them. He's a little more complex, but you're soon going to see that it's not that bad. They've got three compartments two compartments in him, three in him. And here we go. This is someone's drawing, and I got some real pictures later on, of a chloroplast. See that dark band right there? That's one of the membranes. See the band right there? That's another membrane. And see this right here? That's the third membrane. There's a space in here, and something's happening in that space that's important for photosynthesis. If you remember the mitochondria you sent me, there's just a membrane, and inside you have all the photons. There's just two. This thing here, they still think that the chloroplast might have been a bacterium that somehow evolved like mitochondria did, but it's harder to explain this one. Because bacteria don't have three membranes. They have two. So I'm more likely to agree with the mitochondria amongst the bacteria and went into a cell and took home there. They try to say that for this one, and I'm kind of going, I can't say it's not, but I ain't sure you can say it is. I don't have any proof to say not. All right. This, or this, or this. There's a thylakoid, there's a thylakoid, 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 thylakoid. But see how they're stacked up? This stack of three is called the granum. This is a granum. It's a stack of thylakoids. This stack of five is another granum, which is made of five thylakoids. There's another one, another granum, which is a stack. Now, 1, 2, 3, 4, 5. I can't say 5 granums. What's plural for granum?
Granum.
Grana. You drop the UN and I have 5 grana. And the most any grana has would be 5 thylakoids. The least would be 2. And of course, this is the space inside, and there's that third membrane. There's a space inside, and something happens in here too. A lot going on in the chloroplast. A lot more going on here than you saw in the mitochondria. I guess it's harder to make it than to tear it up. What's harder, baking the cake or eating the sucker?
Making it.
Sure, it's going to take more to make the thing than to use it. So I understand why you got more machinery here to make that cake than you have in mitochondria required to break it down. That makes perfect sense to me. I have no problem with that. All right. This is another picture by another source, and it's showing you the same thing I just showed you. And now, see that connector right there? That's the lamella. Now, if I put an E on here, for how many? I see 1, I see 2, I see 3. I see 3, right? I wouldn't call them lamellas. They call them lamellae, with the E on back of it. These are Latin words, or Latin plurals. So in this thing, I see one there that looks like 2. This is like your bridges. So they're kind of connected together. And this one here, this way the bridges are in purple. And again, there's your 5 thylakoids. If I have all 5 circled like I do here, but that's a granum.
Cranium.
There's like a little walkway to get across, and they're all interconnected. That's your lamella. Now, this outer, there's that green band on the outside. That's your outer membrane, and there's your inner membrane. There's your inner membrane. There's no fancy name for this. It's just called inner membrane space. I'm amazed there's not some big bad word for it. And this, of course, inside here, things happen. This, it's a stroma. S-T-R, stroma. So, do I have any label? Oh, yeah, I do label it. Good. The light area. Did I label the other one? Did I miss one? Now, here, your stroma. This is a fluid. So, some things happen in the stroma with the R, and some things happen in the thylakoid, and together they do something called photosynthesis. So, that's all the parts of chloroplast. And it is 3 membranes. 1, 2, 3. That's one that's for real. Of course, looks nothing like that pretty, but you can still see, if you look carefully, there's the outer membrane, and there's the inner one, and there's the inner membrane space. Now, you see this right here? That is your granum. Now, you see the areas here and here where they connect together? That's the lamella. Well, that is a chloroplast. And it's not green because this is taken to an electron microscope. We can't color things in that thing. There's another picture of one. It is not that one, it's another picture. But as you said, they're very similar, and the name again, if you look carefully, there's the outer membrane, there's the inner membrane, and there's the membrane space. The dark areas are the thylakoids. Actually, this whole area is the granum, right? I don't know, I can't count them all, but obviously there's maybe 3 times as much there as there is here. And the light areas between them, that's the lamella. Pretty simple structure, really, though. Pretty simple structure. Now, stages, let me see. I'm not going to go in here. Let me look ahead. I think I'm stopping. I want to start this on a new day. Would you mind? I really feed you in small bites. And right now, I think you're following me. If I go too fast, I'll soon make a dust storm and you'll be lost behind me. So I'm going to make a note to myself. Well, you make a note on your, so I know where to start again. Because I got all these classes going now, driving me nuts. So make a note that we need to start on slide
24.
Actually, I call it 14. See, this number here, I'm going to change that. I call it 14A. We finished 13C, I need to start discussing, and it's a good point to stop. Good point to stop.